The final module, examining the causes of pollinator declines, will discuss the important role of nutrition in pollinator health and how this may be used to increase the overall health of honeybee populations. Don't forget about the quizzes. This is how you receive credit for this Advanced Master Gardener pollinator training. By the end of this module, you will have an understanding of the various building blocks of life, why colonies procure food to fulfill developmental needs, the value of diverse food resources, and differences in nutritional needs of larvae and adults. Returning to our figure on causes of honeybee declines, we have covered parasites and pathogens and pesticides and damaging agricultural practices. Issues that may arise as a result of beekeeping practices were covered in modules 5 and 7. For now, we'll focus on how declining forage and a resultant loss in nutrition affects honeybees. But first, I'll briefly go over the organic components necessary for life. Starting at the most basic level, nucleic acids are essential for all forms of life. These contain genetic materials as either DNA or RNA that code for all aspects of an organism's biology. We describe DNA as being made up of 4 different nitrogenous bases represented by the letters G, C, T, and A. The central dogma in biology describes how we go from DNA to proteins. Inside our cells, the G's, C's, T's, and A's of our DNA get rewritten into a slightly different form or transcribed into messenger RNA. In this case, the T is replaced by a different nitrogenous base represented by the letter U. When these 4 letters come together in different combinations of 3, they code for one of 22 different amino acids, 20 of which are found in honeybees. In this example, the original DNA coded for the amino acids methionine, isoleucine, and serine. Each amino acid has its own particular chemistry, meaning they react differently in relation to each other. Amino acid chains are referred to as polypeptides, and the order of the amino acids within the chain is what causes proteins to fold into unique 3D shapes with a specific function. Any genetic differences at the DNA level that cause substitutions of one amino acid for another could lead to incorrect folding and therefore a potential loss of function of that protein. If this protein is an enzyme responsible for aiding in digestion, for example, then the organism wouldn't be able to correctly digest and absorb nutrients from food, causing death. The diagram here is showing the honeybee protein phospholipase A2, which is one of the proteins in honeybee venom that causes pain. If a genetic mutation changed the folding pattern of this particular protein, it might mean that it no longer causes pain after a sting. In this case, I might not learn that stings are painful and to avoid honeybees, and instead go into their hives whenever I want for some honey. This is an extreme outcome, but you get the idea of why a mutation at the most basic level of our biology could have significant impacts on the life of the fully formed organism. Another building block of life is lipids. These are compounds that are not water-soluble and can include biological oils, waxes, and steroids. Beeswax, produced by wax glands on the underside of the honeybee's abdomen, is a lipid. Carbohydrates are another important building block, and include sugars, starches, and cellulose, and are produced in plants. When an animal eats and digests carbohydrates, they break down into sugars, which are metabolized to produce energy. When you consider embryonic development from egg fertilization to emerging adult, or a jumble of genetic material to a complex organism, how do we go from submicroscopic to visible with the naked eye? Where does all that extra body mass come from? Living organisms, including honeybees, use the essential building blocks of life, with their DNA mapping out where everything goes as the embryo develops, to synthesize their bodies with functioning organ systems. And all of these essential components are acquired from food. When the queen lays an egg, she's depositing a fertilized cell, if it's a worker, of course, as well as nutrient-dense proteins to nourish the embryo from fertilized cell to larva. Think of this as the egg white part of the egg. Nurse bees feed larvae royal jelly, a protein-rich substance produced by their hypopharyngeal glands, and they get the protein to do this by consuming pollen. As flower-feeding organisms, honeybees must acquire all their essential nutrients from nectar and pollen. Nectar, for the most part, is a carbohydrate source, while pollen is the major contributor of proteins and lipids. Of course, honeybees aren't consuming raw pollen from the field. They pack the raw pollen into cells and cover them with a layer of honey and secretions to create a slightly fermented product known as bee bread. In addition to being rich in protein, it also contains beneficial macrobiotics. So there was a reason I spent so much time going over how DNA becomes a protein. Perhaps you've heard the term essential amino acids before. In the human body, some amino acids are coded within our DNA, and we can make them ourselves. But others are difficult for our bodies to synthesize, and thus we have to acquire them from food. These are the essential amino acids, or the ones we cannot make ourselves. In animals, 20 amino acids are required to grow and function properly. The development of body tissue, muscles, and glands are dependent upon adequate amounts of protein and therefore essential amino acids in the honeybee's diet. In honeybees, there are 10 amino acids that are considered essential that they must acquire from pollen and nectar in their surrounding landscapes. As far back as 1953, researchers demonstrated that diets lacking just one of these essential amino acids made honeybee growth and development impossible. The importance of diversity in honeybee diets goes beyond just variety and honey flavors. Plants have varying amounts of amino acids in their pollen, and if a particular species is lacking but is the only one available, a honeybee colony may not survive. Honeybees are generalist foragers and rely upon a diversity of floral resources in the environment to meet their nutritional needs, as opposed to some solitary bees, which are pollen specialists. For these 5 plant species, you can see there are differences in the concentrations of essential and non-essential amino acids. Note that sunflower, represented by the yellow bar, had particularly low concentrations of phenylalanine compared to pollens from the other species. Phenylalanine is an essential amino acid for honeybees, so if colonies were located in an area dominated by this type of sunflower, they may have nutritional difficulties without access to more diverse forage. Here, amino acid concentrations were compared for a single floral resource, dandelion, but across a number of states representing different climates and soil types. Notice again the high amount of variability in the essential amino acids, and this for a single flowering species. In laboratory tests, it has been further demonstrated that nurse bees fed only dandelion pollen in the lab were unable to rear brood because the royal jelly they produced was lacking in arginine and phenylalanine and does not contain tryptophan. The names and concentrations of the amino acids aren't important, but this data does help emphasize the importance of floral diversity within a honeybee's diet. These critical micronutrients acquired from food must be present in sufficient quantities for honeybees to thrive. The best way to ensure this is making sure apiaries are placed in areas with diverse forage. It can be easy to get bogged down in the details of the components of proteins and whatnot, but there are practical considerations for what a diet lacking in diverse protein components means for an organism. It means muscles will be underdeveloped. Thus, those bees that do survive to adulthood may be lacking in musculature to forage as well as they could. It also has implications for overall immunity. During periods of pollen stress in the environment, any adults reared will be smaller with shorter lifespans, which interferes with the colony's ability to acquire resources. It also means the hypopharyngeal glands will be underdeveloped, which has serious consequences for the ability of workers to rear the next generation of brood. Nutritional requirements for essential amino acids and proteins are greater when bees are developing larvae and as young adults, but the requirement shifts to more carbohydrate-heavy diets as the foragers age. Carbohydrates in the form of glucose acquired from nectar, extrafloral nectaries, or honeydew is a metabolic requirement for energy production in honeybees and all other animals. Extrafloral nectaries are just what the name implies. Some plants produce nectar from pores not associated with flowers to attract natural enemies, and honeybees will take advantage of these sugary resources as well. Honeydew is the feces produced by sap-sucking insects, which itself is effectively sugar water and will be collected by tending ants as well as honeybees. From the lecture on pollination biology, you learned about different pollination syndromes and how nectar volume and sugar concentration differs between them. In Melitophyllae, flowers produce a medium volume of nectar with a mid-range sugar concentration. Many Hawaiian flowers evolved with bird pollinators and therefore would be expected to produce large amounts of nectar, but with a low sugar concentration, and therefore be unattractive nectar sources for honeybees. Concentrations between 30 to 50% are optimally attractive for bees. Interestingly, pollen and bee bread also contain 30 to 35% sugar. Nectar is comprised mainly of sucrose, glucose, and fructose, which are different types of sugars. As the nectar is dehydrated and enzymatically converted to honey by the honeybees, it becomes 95 to 99% sugar. After being eaten, a single sugar molecule produces 38 units of energy. So you can see why it is an essential building block of life. Without energy, key activities and behaviors could not occur. Honey is required for overwintering in temperate climates, and a healthy colony may require anywhere from 50 to 80 pounds of honey to survive the winter and early spring buildup. Honey requirements for summer are estimated around 95 pounds per colony, and this is in addition to any honey consumed to produce beeswax, which, if you remember, is 10 pounds of honey to produce 1 pound of wax. While carbohydrates are more of a short-term energy storage compound, lipids are for long-term energy storage in the form of fats, as well as essential precursors for hormones and structural components of cell membranes. Pollen contains anywhere from 1 to 20% lipids. Insects don't have a liver, but the fat body of the honeybee sort of serves the same purpose, and lipids in the diet are important for ensuring a greater fat body, which has been implicated in overwintering success. Therefore, it's important for fall bees to have access to pollen with sufficient lipids to increase colony success over winter. Sterols are a type of lipid and are precursors for hormones that are required for normal growth and development and reproduction. One example is cholesterol, which is a precursor for the production of molting hormones. Without these, the growing larva wouldn't be able to transition between instars and would die, or wouldn't be able to develop properly during the pupal stage. My own research into honeybee nutrition has supported the importance of pollen in boosting larval physiology and the ability of larvae to internally withstand things such as oxidative stress. In a study conducted at an apiary in Baton Rouge, Louisiana, I deprived half of the colonies of pollen at 3 different time points in 2017 using pollen traps, and provided all of the collected pollen as a supplement to the remaining colonies. While the traps collected about 85% of incoming pollen, some still did make it inside. I deprived and supplemented colonies every other day for a total of 4 pollen distribution events, approximating the time it takes for a larva to go from first instar to pupa. Just before pupation for 2 of these events, I collected 5th instars for physiology measurements. May 2017 in Baton Rouge had higher than normal rainfall with lower than average temperatures, which resulted in a pollen dearth throughout the area. Even when colonies were supplemented with pollen during this time, so little was coming into the colonies that larval lipid levels were very low. In contrast, during a strong pollen flow in October 2017, supplementation did lead to an increase in overall lipid levels of the larvae. When measuring a physiological marker for oxidative stress, called superoxide dismutase, I found that even though minor pollen supplementation didn't contribute to overall improved larval status, it did help to otherwise mitigate oxidative stress. This highlights the importance of pollen, particularly during stressful times of the year when weather extremes can alter plant phenology and access to pollen, a concept which is transferable here in Hawaii. But pollen quantity alone isn't enough. Pollen quality is also important to take into consideration, as all plants will produce different ratios of essential amino acids, total protein, and lipids as their reward to visitors. Honeybees, as generalist foragers, must visit a number of different plant species in order to balance the nutrient intake of various pollens. Ongoing research in my lab is identifying and evaluating the plant species foraged on by bees across the islands, something which has never been done before in the state. Remember, honeybees are an invasive species in Hawaii, and plant systems in the state evolved in the absence of honeybees and other social pollinators. This means the nutrient ratios in their pollens may differ from optimal for honeybees. If you recall, Royal jelly is produced in the hypopharyngeal glands of nurse bees and regurgitated into cells for the developing larvae. The quality of this bee product is therefore a direct result of the nutrients acquired by consuming local pollen. I collected royal jelly in February 2019 during queen rearing across the state and found that while total protein did not differ across the islands, lipid levels were significantly lower in royal jelly from an apiary on Oahu and higher on Kauai, though there was a lot of inter-apiary variability. Beekeepers at the time of collection self-reported flowering of variable wildflowers and crops, including avocado, citrus, and macadamia, during the collection period. I would like to follow up on this preliminary study to quantify the exact nutrient ratios of bee-collected pollens and identify those pollens to determine whether honeybees take advantage of native Hawaiian plants or prefer introduced weedy species, and whether nutrition could be driving these choices. The nutritional requirements of immature honeybees differ from those of adults, which is most obviously demonstrated by the fact that they eat completely different foods. Developing larvae are fed either worker or royal jelly, with workers being switched to a diet including honey and pollen before pupation. Just like human babies, they have a need for a diet rich in lipids and protein to fuel growth and development, though to date, no satisfactory diet has been developed to allow complete larval development in the lab. Once adult bees emerge, they consume large amounts of pollen to continue stimulating gland development as they take on the role of nurses. However, as they transition to foraging, their protein requirements decrease and their diet shifts to mostly carbohydrates in the form of honey to fuel flight. Laboratory testing has shown that honeybees possess a remarkable ability to regulate their nutrient intake, though they either do not or cannot distinguish between pollen nutritional values, and therefore cannot communicate information about pollen quality to foragers who are regulating which pollens come into the hive. This is why access to a diverse diet is so critical for honeybees. Having many of a single resource that is balanced in amino acids and lipids would be fine from a nutritional standpoint, but that is not likely to be the case. We can assume that a maximal diversity of flowering resources will dilute any individual plant species that may not be as balanced in nutrition for honeybees, such that the entirety of what they bring back to the hive is optimally balanced. When honeybee habitat is lost as a result of mowing, herbicides, or monoculturing practices in agriculture, the nutrition available to colonies is greatly reduced. Similarly, many landscaping plants in urban areas have no nutritional value to pollinators because they have been bred for showiness and as a result no longer produce nectar or pollen. Considered choices when introducing flowers into the landscape or your own garden is important for maintaining healthy pollinator populations. There's one more bee product related to health and nutrition that hasn't been discussed yet, and that's propolis. Honeybees will collect tree resins and mix these with beeswax and saliva to create this bee product, which they use as a sealant and glue inside the hive. It varies in color depending on the botanical sources from which it originates, but may be red to green, with dark brown being the most common. Propolis is also antioxidant, antibacterial, and antifungal, and can therefore be essential in promoting colony-level immunity even though it is not directly consumed by the bees. Although the colony is comprised of thousands of individuals, genetic studies revealed that honeybees actually have far fewer genes related to immunity, meaning that an individual bee's immune defenses are lacking. It is thought that they overcome this with behavioral strategies to combat disease via social immunity or cooperating to protect the colony with strategies such as removing dead bees, grooming, hygienic behavior, and the gathering and use of propolis within the hive. It can even be used to mummify invaders that die but are too large to be removed. It is not uncommon to find mice or lizards completely entombed in propolis. This prevents the carcass from smelling or decomposing in such a way that would be harmful to the colony. This concludes the section of the course examining causes of bee declines. In order to receive credit for the advanced certification, please complete Quiz 9 found under Lesson Materials. The next lecture will cover the theories and practices for how best to construct pollinator gardens.